Factors affecting enzyme activity. The most important for undergraduates are effect of temperature, of pH, effect of substrate concentration, and enzyme concentration. And most of you were able to answer that you obtain this shape of the curve when you plot temperature against the velocity of the reaction. That also means the enzyme activity. And you obtain this shape of the curve called bell-shaped curve when you plot the velocity against pH. And then there is a rectangular hyperbolic curve when the substrate concentration is plotted, and a linear curve when the enzyme concentration is plotted against velocity of the reaction. So most of you are able to answer this, but purpose of this lecture is to explain why these shapes of the curves are obtained. First, we talk about effect of temperature. As you can see, as you raise the temperature, there is increase in the velocity of the reaction. Why does this occur? The purpose of the lecture. Because with increase in the temperature, the kinetic energy, the kinetic property of the reacting molecule increase. They start moving more fastly, they collide more fastly, as a result of which the enzyme activity, the velocity of the reaction, reflected of course by reflecting of course enzyme activity, that increases in a almost linear fashion. So much so that with every 10-degree rise of the temperature, the enzyme activity is almost doubled. That value is called Q10, which is 2 for most enzymes. But this does not go on indefinitely because at about 40 degrees Celsius or so, the rise in the temperature paradoxically causes sharp fall in the velocity of the reaction. The reasons are very simple to understand. The three-dimensional shape of the molecule is disrupted. The process is called denaturation. So the enzyme protein undergoes denaturation, as a result of which the enzyme activity falls very sharply in a precipitate manner. So initially, rise U10, I explained, the activity reaches maximum, and this temperature is called, the corresponding temperature is called optimum temperature. And most enzymes, उनका optimum temperature कितना होगा? The physiological, our body temperature, 38 degrees Celsius, 98 degrees Fahrenheit, whatever, please correct it. Now we come to the second factor. This is the bell-shaped curve, which most of you are able to very correctly answer and draw. But what is the reason? Much like every enzyme has got an optimum pH, every enzyme also has got much like, we said, every enzyme has got an optimum temperature. Every enzyme has got an optimum pH also. At this pH, the enzyme activity is maximum. The velocity is maximum. But as you can see here, slight change in the pH on the higher side or lower side results in very sharp fall in the enzyme activity. Why does that happen? What happens at the optimum pH? Let us understand it first. At the optimum pH, the substrate and the active site of the enzyme, they are appropriately charged. We say it appropriate state of ionization. What do we understand by this term? Let us see. You people know very well that charge on an amino acid alters with change in the surrounding pH. So अगर pH 7.4 से आप change कर दो, the charge state of amino acid present at the active site of the enzyme, that will also change. It is possible that some enzyme, some amino acid which was present in the positive state at 7.4, change in the pH, it may become even negatively charged or neutral. So when it was in the positive state at optimum pH, it was able to bind with the substrate appropriately. Probably because substrate के अंदर positive, negative amino acids are. So the change in the charge state at active site will influence binding of the substrate adversely. As a result, they will not appropriately bind, and result will be that the enzyme activity will fall. Now, one or two points. So we have tried to explain reason behind bell-shaped curve and reason behind when temperature is plotted against the velocity of the reaction. Just one or two points. Why acidosis, fall in the pH, or alkalosis are so very dangerous. The reason, bell-shaped curve, sharp fall in the enzyme activity. So अगर बॉडी के एंजाइम ही एक्ट नहीं करेंगे, it's a dangerous, perilous state for the body. Second, why in fever the BMR, basal metabolic rate, rises? Answer you can get from this part of the curve. Fever के अंदर velocity of the reaction, enzyme activity, that increases. As a result, reactions become more faster. Second point related to temperature, why we are not able to tolerate temperature beyond 40, 42 degrees Celsius. The reason, sharp fall in the enzyme activity because of denaturation beyond this temperature. And in fact, we tolerate hypothermia in a much better way compared to hyperthermia, because in hypothermia, as you can see, the fall in enzyme activity, rate of the reaction so-called, that is not abrupt, that is very gradual. So 0 degrees Celsius also can be tolerated with practice, with patience. Uh, so I think we have discussed two of the factors. Now we will be talking later about substrate concentration and enzyme concentration in some later video.